Hello, my name's Tom and I'm a research scientist at the University of Aberdeen working in the Institute of Medical Sciences. Today I'm going to tell you about how animals build themselves. You may not have thought about this before, but it's a really remarkable process that we all did when we were developing. Now I'm going to show you two different species of animal today. The first you're very familiar with, humans. This is an example of a human, that's me working in a research laboratory. And the second species of animal, and I'm actually clutching the fish tank that houses this animal in the left-hand photo, is what's called the zebrafish. And zebrafish are a really excellent animal to study because they're small and they're transparent. More on zebrafish later. To start with, how do humans build themselves? Well, this is not an obvious question to ask. One of the most amazing things about life is that all animals, including humans, start life as a single cell. And this is remarkable. That single cell, it's very humble, it's very simple. It's just essentially a blob with some DNA in it. This blob transforms into a very complex creature that has ears and eyes and hair and arms and legs, hands, fingers. It can see, hear, taste, touch. It can move, it can decide, it can feel emotion. And it is incredible to think that all of this complexity has emerged from something so simple. Now, the process by which you go from this simple single cell to a complex animal is termed development. And for us, this happened when we were in our mother's wombs. Another one of the really amazing things about our development is that we started off absolutely tiny, less than a millimeter, much less than a millimeter in diameter. And if I was showing you this to scale, we wouldn't be able to even see how big we were when we started out. To give you a sense of the perspective, imagine if we were a single cell and we were a single cell that was the same size as a pound coin. Now this single cell increases drastically in size. If we started out the size of a pound coin, we would now be the size of a very, very large skyscraper. This is one of the tallest buildings in the world. It's called the Willis Tower. It's in downtown Chicago in the USA. And it's a staggering 440 meters approximately high. It's amazing how embryos not only increase in complexity, but increase in size. Going deeper on the example of the Willis Tower, you might appreciate that to build this very impressive structure, we needed many different people and a lot of expertise. We needed architects to design the building, specialized people to load the glass, a whole host of builders, plumbers, electricians, and project managers to coordinate the entire assembly and processing of this final impressive building. However, in contrast to all man-made structures, the amazing thing about animals, and that includes humans, is that we must build ourselves. All of the instructions contained about how we build ourselves is within the single cell. It's within the DNA of that single cell. And without the help of engineers or architects or plumbers, we spontaneously transform from a single cell by ourselves into a complex animal. And it's this amazing process that I, together with many researchers around the world, study. So how on earth does it work? We have a few clues as to how it works. And in particular, we know of two very important processes. The first process is something that you did as the first thing that you did in your life, and that is that you divided. You started as a single cell and then you divided into two. And I'm going to show you an animation of what we think an early human embryo looks like. We start as a single cell, but then divide to create two cells. And we do that again. Each of these two cells divides to create another 2 cells to make 4, and then again to make 8 cells, and then again to make 16 cells. And this process keeps on going and keeps on going until much later in development we have thousands, if not millions of cells in our embryo. So step 1, divide. Step 2, move. When you have all these thousands of cells, they must move around to create different structures. And again, I'm showing you an animation of a human embryo, now at a later stage, and we're looking at the formation of some of the earliest structures and organs of a human. Here, we are zooming into the very heart of the embryo through the developing spinal cord. You'll see the spinal cord is curling up on top of us, and as we zoom out of the embryo, we can see these 2 sides of the brain starting to form. We can see, if you look closely, the formation of the ribs, or the precursors to the ribs. We see in red already a beating heart together with blood vessels that go around the body of the animal. We can see the eye, and here is the brain of the early human. And so relatively quickly we've gone from a very simple structure to something that starts looking like an animal. And this process is then extended to turn this early human into a more adult and fully formed human. But these are just animations. They're our best guesses for what's going on in a human. But can we watch the real thing? Now, this is impossible in humans because all of this is going on inside of the mother. But perhaps we can watch it in different species of animals. But there's another challenge. Embryos are tiny, and I've already told you this, but just to illustrate the point, imagine if a pound coin was this big, we still couldn't see an embryo. We'd have to zoom in to a very small bit of the pound coin, and then we can start faintly seeing the size of a single-celled embryo. So in order to be able to really understand and watch embryos develop, we need high-powered microscopes. Microscopes are kind of the opposite of a telescope. A telescope is used to look at the stars, really distant objects. Microscopes are used to look at very close objects that are very, very small. And the objects that I look at are tiny zebrafish embryos. So now we're coming back to the zebrafish. Why zebrafish? Why is it useful or interesting to study how zebrafish develop? Well, really, it's because they are very similar to us, particularly in the ways that they develop. And actually, genetically, we're very similar. And many of the disease genes that cause defects in humans, particularly those associated with the errors in the growth and development of an embryo, they have exactly the same functions and disease manifestations in the zebrafish. So by studying the zebrafish, we might learn some very interesting things about how humans develop. But the real advantage of zebrafish is we can watch them live. So I'm going to show you a movie of one day in the life of a zebrafish embryo, and it turns from a very simple structure into a very complex one. So this embryo is 30 minutes old, and this time is going to change. You will see that the cell is highlighted here in yellow. It's a very simple structure. Now, humans, we don't need anything else. We get all our nutrients from our mum. However, many other animals have what's known as a yolk, and this provides food to the developing embryo. You'll be familiar with yolks for chicken eggs, for example, that you would buy in the supermarket. So the cell, the embryo proper, is sitting on top of its food source, the yolk. Now, if I play this movie, you will see this single cell dividing many, many times, giving rise to a pulsating mass of thousands of cells. If you look carefully, you will see these cells are starting to move over the yolk, But still at this stage, there is not much structure. Then, at about 9 to 10 hours, the magic happens where we start to see things. We start to see organs forming. On the left, you'll see the eye starting to form. This is the brain. You'll see the ear starting to form here. You'll see the equivalent of ribs starting to form along the length of the fish. And you can see that the tail of the fish then starts to form. You can see a beating heart beating away here, and it's pumping nutrients and oxygen around the whole length of the animal. And within a day or 2 days, we have something which is very complex. And if we hadn't anaesthetized it, would be feeding, moving, and responding to stimuli. Now, if you remember the 2 key steps I showed you about human development, they're exactly the same in zebrafish. Step 1, cell division. Here is a zebrafish embryo undergoing cell division. And we can really see at a high resolution, we see these cells are dividing, almost seething with activity. Then, much later, we have quite a simple structure, and then all these cells have to start moving around to generate organs and structures. And it happens quickly. This is just a few hours. We see the eye here is forming. We can see along here the ribs starting to form. And the tail is slowly growing outwards towards the end of the animal. Up here you can see the brain, and then also what is going to be the ear of this fish forming. And this is taken less than a day to happen. It is a remarkable process. And it is my hope that by studying with very high-powered microscopes how this happens in zebrafish, we'll understand better how it might happen in humans, and thereby understand why it can sometimes go wrong in human congenital defects. So I hope that's given you a little flavour into the remarkable process of embryogenesis and animal development. Thank you for listening. Goodbye.